Hi, my name is Dirk Valkenborg, and I'm from Hasselt University. I'm a partner of the Beyond Genomes project, and this is a first tutorial in an entire series where we try to explain something about proteomics, mainly mass spectrometry-driven proteomics. So there is an entire branch of covered by proteomics. There are all different types of proteomics. What we will focus on in this series is discovery proteomics. So that means that we will analyze a sample of a patient or a plant or any organism, and we will try to identify and quantify as many as possible proteins that are present in that sample. And we will try to do that with mass spectrometry. So discovery proteomics is the topic of this series. And as already I gave this lecture multiple times and also in courseworks, and what students say afterwards as well, it seems that there is much noise in the data and few signal. And that's exactly true. And that's why I have this cartoon here on the slide, which is an indication that actually looking for discovery proteomics is not looking for a needle in a haystack, but a needle in a stack of needles. So it's a complicated technology that's used. It's a complicated biological framework that we're in. And in terms of algorithms and bioinformatics, it's also complicated. So it's 3 times complicated. And I will try to do in 1 or 2 sessions, give you a brief introduction about what is the surrounding biology context, what is the technology context, and how will we now analyze the data, which types of algorithms will we use to analyze the data. And there are some pitfalls that we should mention, and that's what I will do probably in the next series of this tutorials. Okay, so what is proteomics? I put a definition here on slide, and proteomics is exactly the same as genomics. So it is the same ID. Proteomics is the direct qualitative and quantitative analysis of proteins in cells, tissues, or organisms under a given set of physiological and environmental conditions. So what you want to achieve here is a direct analysis of all the proteins at once. So, and we want to know how these proteins interact with each other and with their environment. For example, if we administer a new pharmaceutical to a patient, then we're interested in how this pharmaceutical will interact and change, for example, an entire proteomic pathway. Instead of particularly studying one protein and their interaction with the drug, we want to analyze all the proteins in a given set of conditions. So we have genomics or transcriptomics. So we have a variety of omics disciplines, and most famous are transcriptomics, genomics, and we analyze genes or gene transcripts nowadays with next generation sequencing and microarrays. Proteins, proteomics, or also the metabolomics and the lipidomics. These are typically analyzed by mass spectrometry. There are some changes nowadays where proteins are also being analyzed with what I called closed box solutions, like Olink proteomics and somologic proteomics. I will not discuss these technologies in this series. So we will only focus on what I call open box solutions, and that is mass spectrometry. When I say closed box, open box, then I mean with mass spectrometry, we don't know exactly what we're looking for. And with these closed box solutions like Olink and SomaLogic, we know the proteins we're looking for. So we need to, that's more like a targeted approach versus an untargeted approach. So a consequence here is that when we do mass spectrometry-based proteomics, we don't know what we're looking at. So we need to identify our proteins first. And one of the paradigms in proteomics is that we want to identify first before we quantify. There are other paradigms where quantification is given priority, but that's something we will not discuss in this series either. So we need to first come up with an identification. And that's what we will do in this series. So go through the biology, go through the technology, and see how we can learn how we can identify proteins. This for people that are have an experience in bioinformatics, I already can give a heads up here. So the algorithms that we will use to have protein identifications, they are more or less similar than the algorithms we use for alignment. Because when we want to retrieve a certain gene sequence from a database, and this gene sequence is obviously altered by biological processing, and we want to have a gene that is quite homologous to the gene in the database, then we need to have an error-proof alignment algorithm to retrieve the genes. So that branch of bioinformatics on sequence alignment, that's also something we will do or want to do with proteins. The only difference is that we are not aligning in the text domain, but we're trying to align stuff in the spectral domain. That will complicate things tremendously. So, but don't worry if you don't understand this completely. We will further explain this further on the slides. So let's first have a look at the biological context. So what defines life's complexity? So in the Beyond Genomes project, we ask ourselves the question, we all know that DNA contains enough information to identify a person, but the question is the same information there in proteins? And probably, we'll see later, the answer to that would be yes, but do we have sufficient technology to find these differentiating features between persons? But let's first look at what defines life's complexity. So if I look at this C. elegans soil nematode that has approximately 23,000 genes, and I compare that with Homo sapien, so us who have 23,000 genes, well, then the differences in the number of genes does not explain to me the differences in complexity of the life form, because I consider a human a little bit more advanced life form than a soil nematode. So I don't think the number of genes do necessarily count as a differentiator here. So what would then define the complexity of life? Well, first of all, there are various levels of molecular organization. And we know all the dogma of life. It all starts with DNA. DNA is our map, but proteins are the world, right? So DNA will be transcribed into a messenger molecule, a messenger RNA molecule, because DNA is way too precious to be used as a worker molecule for making proteins. So that's why DNA in the core of cells are transcribed into pre-mRNA molecules. And these mRNA molecules, they can undergo processing. And alternative splicing is one of these processing forms. And from one simple DNA gene, we can have multiple mRNA molecules. So you can see here that we already have a combinatorial expansion of possibilities here, and see how one gene can result to multiple mRNA molecules, and these mRNA molecules in turn will be translated into proteins. We'll see on the next slide. But just to give an example on how much isoforms, protein isoforms, a certain gene can give is, for example, here in the DSCAM gene that contains 24 exons, and then potentially could give rise to more than 38,000 different protein isoforms. So that's one gene that can translate into more than 38 different 8,000 different proteins, and that's a lot. So we're not there yet. I think the next level is where we will translate the mRNA molecule into a linear protein. That's a straightforward process because an mRNA codon translates for one of our 20 amino acids, and we will have obtain a linear sequence of amino acids, and then some magic will happen, and life is self-assembling. So these linear molecules, they will start folding and adhere to the principle here as maximum energy, minimal entropy. So they will fold into proteins, and then they will be matured, dressed with some functional groups so that they become functional proteins. And that dressing, so during the life of the protein, a protein can undergo multiple modifications, and we call these modifications post-translational modifications. So these are like, you could see it like some handles and grips that can be attached additionally to proteins, and that can define then the function and the purpose of that protein. So protein, where the backbone can be the same, the linear structure can be the same, but depending on the modifications, PTMs, the post-translational modifications in proteins, it can have a different function or can have a different localization in the cell. So all these PTMs will lead to, protein isoforms, obviously. And we'll have more possibilities of proteins. So what is even more interesting is that these proteins themselves, they start to interact with each other, as you can see here on the slide. So the interaction between proteins, they can form complexes. And some basic units, some proteins form a basic units and can be reused in different larger protein complexes. So you can really see this as well, nano machines, biological nanomachines that are constructed in a cell, and depending on the assembly of a protein in a complex, the function of the protein can differ. So it is a complicated matter, proteomics. Okay. So if you then look at these various levels of molecular organization, then yes, indeed, we have 23,000 genes in our body. But when we consider the transcriptome and the proteome, we always almost arrive at a billion different protein isoforms. So this is why proteomics is way more complicated than genomics. Also, the molecule is different. So genome, nicely linear molecule that is composed out of only 4 letters, right? A, C, G, T. So nucleotides, 4 nucleotides. The proteome, this is 3-dimensional nanomachines with as basic building blocks, the 20 amino acids. Same slide, different slide, same concept here. So here, one gene can give rise to a lot of protein isoforms, right? So the possibility here is if a clinical, so the gene might be the same, but maybe the clinical status, we have a patient that is a normal, healthy human, a human with cancer, and a human with a benign cancer. So they could all have the same gene from genome perspective. There's nothing to differentiate between the clinical status, but from the proteome perspective, it could be possible that one of the proteins would carry a different post-translational modification, and that could then serve as a biomarker to early detect cancers, for example. So I hope I convince you guys that that the field of proteomics is a challenging domain. It's a complicated domain, and it's worthwhile because when looking at proteins, we're so much closer to the phenotype, actual phenotype, than when we would look at the genome. Okay, so which technology are we now going to use to detect all these PTMs on the proteins? Because that's our ultimate goal, finding proteomic fingerprints or biomarkers that are predictive or diagnostic for some clinical status, or maybe to have personalized medicine, for example. So some patients would benefit from some therapies better than other patients. So that's what we want to achieve here with this discovery proteomics. The technology that we're going to use for this is mass spectrometry. This is a pretty old field. The mass spectrometer was only invented in 1913 by J.J. Thomson and W. Aston in Birmingham. Read about the history of mass spectrometry on the wiki page. It's a fascinating story. But if you look at the date, it's a bit older than, well, it's a bit longer than a century ago that mass spectrometry was invented. And in the beginning years, mass spectrometry was not really used to analyze proteins because proteins are way too big molecules to be ionized, because we needed to ionize our molecules before we can analyze them with mass spectrometry. And mass spectrometry was therefore more the play field of the people that were investigating atoms or metals, heavy metals, for example. And that's exactly what J.J. Thomson was doing. So he was looking at beams of positive rays of neon, and he discovered for the first time that neon appeared in nature in two variants, and that was also the discovery of isotopes. So that's isotopes were the presence of isotopes. Apart from radio isotopes, obviously that was known, but naturally stable isotopes, that was not known before 1913. That was a discovery made by the first mass spectrometer, and neon was discovered to have two isotopes. So time passes. We have 1950, Watson and Crick that discovered the structure of DNA. And the whole genomics field started to boom. And by the 80s, we got technologies that were pretty able to. I mean, we had a good hope for genomics to become a high throughput discipline. And nowadays, genomics is a high throughput discipline. So today we're at the moment that it's maybe cheaper to resequence a genome than just to store that data on a server. So it becomes a common technology. But we didn't have that hope for proteomics because it was way too complicated. Only in the mid-80s, there was a new invention of soft ionization that suddenly allowed us to use mass spectrometry to study proteins. So only in the mid-80s, mass spectrometry entered the field of biology. Before that, there was a Edman degradation or some very complicated analytical chemistry techniques to try to figure out which proteins we're looking at. One PhD, the identification of one protein would cost one PhD, for example, but that changed in the mid-80s. And the soft ionization techniques that I'm talking about are MALDI. Sorry for that. It's MALDI ionization. It was discovered by Tanaka, and it's electrospray ionization discovered by John Fenn, who passed away recently. So these two ionization techniques, they allowed us to ionize proteins or peptides without breaking them during the ionization phase, and that's important. Okay, so small anecdote: there was a Nobel Prize awarded for these two ionization techniques. So later on, so that to just illustrate the importance of that. So good, we can ionize proteins now, and there are different branches actually of mass spectrometry that study proteins and peptides. In this series, I will only focus on one particular branch, and that is called shotgun proteomics, also known as gel-free proteomics, or also known as peptide-centric proteomics, or also known as bottom-up proteomics. That's what we're going to do in this series. And we will focus on a particular technological setup. So there are different technological setups we could use to do shotgun or gel-free proteomics. I will only limit myself to one particular setup, and I can explain that here below. So in a typical shotgun proteomics setup, we have a technological device called chromatography. I will explain that later. We have an ionization step. I will explain that later as well. We have a mass analyzer, we have a fragmentation step, and we have a detector. And for each blue block here, you can put numerous technologies, and you can make hybrids of all types of and combinations of all types of technologies. And these different combinations can be used for different purposes. So there is a lot of variety in the technology used for shotgun proteomics. I will limit myself to, well, I think the technology that I will describe here is perhaps 70% used for this type of shotgun proteomics. And I will use reverse phase nano-LC. I will limit myself to electrospray ionization. As a mass analyzer, I will use a kingdom trap or an orbital trap. For the fragmentation, I will limit myself to collision-induced dissociation, and for the detector, I will use the field. So that is the setup I will discuss here. And do note, there are a lot of hybrid mass analyzers today. And well, this is just I can have here for each blue block. There are maybe 10 competing technologies that have 10 different characteristics and will lead to different type of data sets. So if you think after this series that you know everything about proteomics, you're wrong. You just know one particular flavor of technology in the context of shotgun proteomics or bottom-up proteomics. So why do I call it bottom-up proteomics? That's because in this technology, we will not analyze the proteins in their intact or native form. So we will actually not try to identify the protein. We will try to identify the protein by their peptide homologies. So what does that mean? So because this protein is too big and too big to ionize and too difficult to handle, we will chop that up. We will chop up the protein in smaller parts, and these smaller parts we call peptides. So we use a molecular scissor to cleave our protein into smaller parts. And these molecular scissors, well, are called enzymes. So they cut, they cut or digest a biomolecule, and in this case, these enzymes, they digest proteins, and we call them proteases. And we use those proteases to cleave our protein into smaller peptides. And it does this by breaking the peptide bond. And it is very specific. It can be, there are different types of proteases, and they will all cut at a very specific site. Just as a reminder, a protein approximately can be composed out of 35 to 50 peptides, right? And the peptide is approximately 10 to 15 or maybe 20 amino acids long. So instead of looking at proteins, we will now try to look at peptides, and we will try to identify the amino acid sequences of the peptides. And when we know the amino acid sequences of the peptides, we will try to interfere which proteins are present in the sample. So we will not directly analyze the protein. We will do this by means of the peptides, and that is a first complication of the technology, because we don't know which peptides. And so when we analyze the peptides, we don't know which belongs together, right? So it's just a bunch of peptides, and we don't know anymore from which protein exactly. We need bioinformatics to solve this puzzle. Okay, so some definitions here. So you see here is a peptide. So typically we present peptides from the left to the right. At the left side we have our amino acid termini. On the right side we typically denote with the carboxyl termini, and we call this an N termini or a C termini, right? So that's how we indicate the peptide. If we cleave a peptide at the peptide bound, a new C terminus and a new N terminus will be formed. Okay. I will go quickly over this slide. All life forms are on this planet are constructed out of these 20 common amino acids. There are only 20. We can group them based on some characteristic. We have aliphatic amino acids, we have aromatic amino acid, acidic, basic, hydroxylic, sulfur-containing, only 2 sulfur-containing amino acids, methionine and cysteine here. Amidic amino acids, we have non-essential amino acids. That means our body is capable of making that amino acid herself. And we have essential amino acids, and that means that we need to eat them. So and that's interesting. So you see there are some essential amino acids like isoleucine, leucine, valine, lysine, depends its organism specific. So what is essential or not? But these amino acids, we need to eat them to have them in their body. Our body cannot make these amino acids ourselves. So, and that's it, what I want to say about these 20 amino acids. Of course, they have a long name. Here you see the one letter abbreviation, and you also see the 3-letter abbreviation on this slide. And below the 3-letter abbreviation, you can see the nucleotide codon that encrypts the amino acid in the mRNA molecule. And you can see that one amino acid can be linked to multiple nucleotide codons. So it means that if there is a change in a letter, it doesn't necessarily mean that the amino acid has to change. So that's already a level of robustness that's built into our biological system. So a single nucleotide polymorphism in the genome doesn't necessarily have to lead to polymorphism in the amino acid, single amino acid polymorphism. Okay, let's have a look at the technology now. I've 10 more minutes to go, so probably I will not finish up this lecture, and I probably need 2 more sessions to complete the slide deck. And so the first important step is liquid chromatography. So I told you you don't look at proteins in the intact form. We look at them. We look at the peptides. So we have a bunch of very complicated peptide mixtures. And if we would give that sample to a mass spectrometer, it would choke on this complex sample. We will never be capable of looking at all these peptides. So we need to reduce the sample complexity. And we do that by playing a little bit with the physical chemical properties of our analytes, or peptides in this case. And I always use this cartoon in my classes. So if there is a mission here, you need to go to the Gap store. So you have the Weinigem shopping center in Antwerp. And if you need to go to the Gap store and buy a pair of pants, you could use that task here to separate between two populations, for example. So you have people with onion money, and you have people that do not have onion money. So that's shopping maniacs, right? So, and people that do not have a shopping mania, so they will just go to the Gap store and probably buy the pants and are out of the Gap store in 6 minutes, and they spent $33. But the people that have onion money, that would probably spend like almost a half day in the shopping center and would have spent like more than $800. So this is a way to separate two species from each other based on a characteristic, and that's what we will do also in liquid chromatography. But instead of using a human characteristic in this case, we will use the physical chemical properties of the analyte. So let's look at this more concretely. So I've put a picture here on the slide of a typical liquid chromatographer that you can see in the lab. So what we will do is we have two liquids. So I'm describing here reversed phase chromatography, and we use nanoflows here. So nano reverse phase liquid chromatography. So we have two liquids here, A and B, and one liquid is a bit watery. It's an aqueous solvent, and another liquid is acetonitrile. So it's more like an organic solvent. It's oily, right? And what we do is before we start the experiment, we will put all our proteins on this nano-reversed phase column. So all our proteins are on silica bead column, and they are there dry and safe. So, and we also have a needle, and I will talk about this needle later on, but it's not important now. But remember that all the proteins are dry and safe on this column, and we start to rinse this column with mainly water only. And there are some peptides that prefer water better than the silica beads on the column. So these peptides, they jump into the liquid phase, and they will be rinsed off the column until they arrive at this needle. And and we will do that for a while, and until no more peptides elute from the column. And so peptides elute from a column, and then gradually we will start to change the. We gradually start to change the mixture of the liquid, and we will make it more oily until peptides that prefer that more oily environment are comfortable to jump into the liquid fluid and will be transported to the needle. And this process goes on a long while, as you can see here in the graph at the bottom. This is not a spectrum, but this is a chromatogram. So you can see that this experiment, this liquid chromatogram experiment, can take up to 90 minutes, sometimes 2 hours. So it's a long experiment, right? And the idea of this experiment is to, well, get rid of all the proteins on our column and transport them safely to our needle. Okay. Also note that there is some formic acid, Fa, added to our liquid. That is an acid, and that acid we need to provide protons. And why do we need protons in our liquid? Well, at some point we want to do electrospray ionization. We look at the ions. We want to generate ions, and ions are the fourth state of matter, right? So we have solid, liquid, gas, and ions. So how do we generate these ions? Let's have a close-up look at this needle here. So we have indeed peptides that arrive in our needle. There is a high voltage span between the needle and the inlet of our mass spectrometer. And we also have a barrier, right? So we spray, and so this might be a little bit counterintuitive here. We spray our analytes that we want to measure in the spectrometer. We don't spray it directly into our inlet of the mass analyzer, but we spray it orthogonally, which is weird, because why would we do something like that? We want these analytes to go into the mass spectrometer. So the reason is we have this high potential field, electric field here between needle and inlet, and we have sprayed these small droplets with our analytes in the atmosphere. And what happens is that there is also this form still present, right? So acid is a donor of protons. So, and these nebulas are very small, tiny droplets, and they will evaporate quickly. So what will happen is that these droplets, they will evaporize. They are charged because of the protons are there. So the protons will repel each other, and a droplet will break into smaller droplets. Evaporation will speed up, and then even more positive charged protons. So a proton is a positive charge. Will repel each other and will break up again, so that the droplet, the liquid, will evaporate very quickly, and at some point the proton that is still in the liquid together with the analyte will jump on the analyte, and we have an ion created. And because we have an ion, it's a charged analyte. The ion will follow our electric field and will be guided towards the analyzer, and then it will be sucked up in the vacuum of our mass analyzer. So what happens with the analytes that do not get a charge? Well, they will be sprayed against this barrier. So that ensures that we only have charged ions in our mass spectrometer, and that's very convenient because we need these charges to manipulate ions and ion beams. And in the mass spectrometer, we call this ion optics. Like you will manipulate light with lenses. That's how we will manipulate ions in a mass spectrometry with electromagnetic lenses, actually. And I will conclude my first session here at this moment before I will start talking about mass spectrometry. Okay, welcome to this second session where I intend to give you a quite complete overview of a typical proteomics workflow. Last session, we discussed a part of the technology, and I want to continue with this and move forward to the next component in a proteomics workflow, and that's obviously the mass spectrometer. And a mass spectrometer can be considered as a very expensive balance. So it's a typical price around of a new mass spectrometer that will be ranging between 700,000 and 900,000 euros if you want to have the latest research instrument, or if you want to look into more standard equipment like triple quads that would cost you approximately like 300,000 or 400,000 euros at this moment now in 2023. So a mass spectrometer is an expensive ballast, but what does it weigh? It weighs molecular ions. So it means that it is so sensitive that it can measure the mass of a molecule. But before we can really measure this, we need to ionize this molecule. So that's why you can see that these molecules here are have an additional proton. So they have now a positive charge attached to the molecule, and that is the fourth state of matter. It's a plasma or an ion in this case. So in my slide here, this is charged with protons. So these are hydrogen atoms without an electron, so that makes it positively charged. And it's also possible to have molecules that are negatively charged, so they have an additional electron. So we need this charge to manipulate the ion beams inside the mass spectrometer and do some ion optics, as we call it, inside our instrument to steer these beams of ions throughout mass spectrometer and have the ability to manipulate and measure the weight of these molecular ions. So how does it weigh these molecular ions? It doesn't do this in kilograms. So an ion is way too small to be measured in kilogram. We use the term Dalton, and Dalton is also a measure for mass. And 1 Dalton is equivalent to 1/12 of a 12C carbon atom per definition. So that is the definition of a Dalton. So what type of data is now produced by a mass spectrometer? Well, it's called a mass spectrum. And the typical mass spectrum can be looked here in this graph on the right side, where we have on the x-axis a measure that is related to the mass of the molecular ion. And it's on the x-axis, and on the y-axis, we have an abundance measure. Typically, the length is an intensity measure, and they can go really high. But what we see in a mass spectrometer often is that the largest peak is normalized to 100%. So we see this often happening in the mass spectrometer. Because these intensities alone, they don't tell us that much. And if you are comparing between different technologies, then these intensities are also totally meaningless. But the relative proportions between these peaks, well, that is relevant information, obviously. So that's why often the most abundant peak in the mass spectrum is normalized to 100%. I told you something about the x-axis, that this is a measure for mass. More or less, I was not completely correct here. Actually, on the x-axis, we have the mass to charge. But I will not go into any details here. So what you should remember is that if a molecule has a single charge, it can have more, it can have 2 charges or 3 charges, but when it has a single charge, then you can read from this axis the mass in Daltons. With the subtraction, if you subtract the proton, the mass of a proton from the mass that you measure. So the mass of a proton is also 1 Dalton. That's 1/12 of a 12C carbon atom. Okay. So I move to my next slide. I got this graph from the website of ASMS. ASMS is an American Society for Mass Spectrometry, and you can find on their website a whole bunch of interesting tutorials. And what I like about this visualization is that sorting ions is like sorting some change money that you have in your pocket. So if you are in the US and you have some dimes, pennies, nickels, quarters, and so on, what you need to do is, when you want to know the amount of money that you have in the pocket, you will start sorting these coins by their face value. So you will sort the dimes, you will sort the pennies, the nickels, the quarters, and the half dollars, and then you just count how many you have of each. And then you know the amount of money you have in the pocket. Well, it's exactly the same thing that happens in a mass spectrometer. Only the mass spectrometer will not sort the ions according to their face value, but according to their mass to charge ratio. And for each typical mass to charge ratio, it will then count the number of ions and presents that number as an intensity value. So we don't really have these discrete values of the number of ions in the trap. Instead, we receive an intensity measure. But that intensity measure is obviously related to the number of ions in the trap. Good. So let's have a look how what type of components are existing in such a mass spectrometer, and I will limit myself in this discussion to a typical class of instruments that is quite often used nowadays for proteomics, and that's an Orbitrap. And Orbitraps are often combined with other type of mass spectrometers composing a hybrid instrument. So on the picture here at the right, you will see an LTQ Orbitrap. So that means it's a linear trap quadrupole. That's the first box that you see here. And behind that box, you see the Orbitrap. Now the Orbitrap, it's depicted here. The box is huge, but the trap itself is very small. It might be 2 centimeters on 2 centimeters with a very extreme vacuum inside there. We express that with the measure torr, T-O-R-R, and the vacuum inside such an Orbitrap is deeper than the vacuum we have in space. So very important, because otherwise our molecules would collide with all types of atoms, and they will heat up and they will break apart. And we don't want that. Well, we don't want that at this stage in the measurement, but at some point we will do this on purpose. So let's have a look first how we will measure in the vacuum. So first we have the electrospray ionization source. I already explained that. And then we have a linear ion trap. So we have 4 metal rods that are very precisely engineered, and we apply a radio frequency and a direct current on these rods, allowing to stabilize the ions within these 4 rods. And then we have also 2 electronic gates that capture the ions and prevent the ions from escaping this trap. And then we will apply a sweep over the radio frequency, allowing to eject some of the ions according to their mass charge, and then measure which ions are injected and how many actually. That's what we want to know. In doing so, we produce a mass spectrometer. A mass spectrum, sorry. Well, this has several, so this is a hybrid instrument, so it has 2 mass analyzers, as we call those. There is an additional C-trap, and that C-trap is needed to inject the ions in the Orbitrap, where we also can measure a mass spectrum. The advantage of having these 2 mass spectrometers in one big box is that the Orbitrap has a high mass accuracy, high resolution properties, and generates very good informative mass spectra. So typically we want to have a precursor scan or an MS1 scan of very high quality. However, such a high quality scan that requires some amount of time. Typically that's 1 second, which is not so long in a human life, but for when generating mass spectra, 1 second is already a little bit too slow for us. And there is second type of data that we want to generate. So please note here, I will talk about a different type of mass spectrum now. And that's what we call a product ion scan, or tandem MS scan, or an MS2 scan. And the Orbitrap is a bit too slow sometimes to process this. So that's why very often we see this lower quality instrument, which generates lower quality spectra, but can produce fragments and can produce fragment mass spectra at a much higher speed. So that's the motivation for having these hybrid instruments. Well, having said that, nowadays we've seen also quadrupole Orbitraps, which where also the MS1 spectrum and the MS2 spectrum is actually recorded in the Orbitrap. Now I need to talk to you a little bit more about this difference between the MS1 and the MS2. So the MS1 is, at some point, the ions that elute from the column. You want to know what's going on during the separation process, and you collect you want to measure all the molecular ions that are eluting from the liquid chromatograph. So therefore, you take this high quality recursive scan or MS1 scan. But the mass alone is not really predictive for the type of ion you're looking at. For example, if I have an amino acid of, a peptide that is composed out of 4 amino acids, being my name, Dirk, for example, D-I-R-K, any permutations of these letters would be a valid peptide, but it would boil down to exactly the same mass. So mass alone is not sufficient to differentiate between the different peptides that exist. So we need to come up with something more clever to uniquely identify the molecular ion. And that is what happened during the product ion scan or the tandem MS or the MS2 scan. It is also a mass spectrum, but it's a different type of mass spectrum. What we will do is we will let the instrument isolate some of a type of molecular ions of the same M/Z value, of the same mass value. And we accumulate a bunch of those ions, like thousands or 2,000 or maybe up to 10,000 of these same ions with the same mass. And then we will start to fragment these, and I will later on explain the fragmentation process. But you have to trust me on the word now. These peptides will break and will exhibit a special fingerprint that is predictive for their amino acid sequence. So I hope I now clearly explained how you can discriminate the MS1 precursor scan from the MS2 scan. And because we want to make as much as possible of these MS2 scans, we want a fast mass spectrometer to handle this. And that's why in the early Orbitraps, they were hybrid and had these linear ion traps in front of the Orbitrap. Okay. Good. So I promised you that I would talk a little bit about this fragmentation step. So that is what we call collision-induced dissociation. Only one type of fragmentation that we can talk about. There are many, many different types of fragmentation possible. And every time you look at the different type of chemical reaction or fragmentation, then will give you a different type of data. And you have to be aware what type of fragmentation technology is used to generate this data. So the idea of fragmentation is, again, the same. I will use my shopping metaphor here. So we have a street in Antwerp, the Meer, famous shopping street, during the Christmas period, and there's a lot of people there that wants to buy some gifts. So if I would start running through these crowd or just them, then I would bump into these people and then suddenly bump more and bump more, I will become unstable because I keep on running, and then suddenly probably I will fall at the sidewalk and break my leg. So that's exactly what will happen with this peptide. So the idea is that we will make it unstable by letting it bump into smaller atoms. It will heat up and it will release the energy by breaking or fragmenting. That is the idea of the fragmentation. Be aware here that we are not talking about one molecular ion of a certain mass to charge or mass. We are talking here about 10,000s of these molecular ions that we're able to isolate. So it is a little bit the game of population statistics that we're playing. So let's have a look how this exactly works. So in our mass spectrometer, we have a certain region where we allow some gas inside the vacuum. So the vacuum here is not that deep, and you can see here our nitrogen atoms positioned in our collision cell. They're just randomly moving around there. And what happens is then we have our peptide that we have selected, not only one, but we have 10,000 copies of these guys. It will move through my collision cell, it will heat up and becomes metastable, and then suddenly release the energy by breaking into two parts. And that is that these two parts, how it breaks up, we will measure. Now, interesting to know that such a peptide can break at several positions. So typically, it will break at the peptide bound. So it will behave very nicely in CED. It will always break at the peptide bound, and that is the connection between two consecutive amino acids. So, for example, consider again the peptide Dirk, D-I-R-K. So it can break between the D and the I, between the I and the R, and between the R and the K. And while breaking, it will produce fragments of a different mass. And it is those fragments that we will measure. And based on that sequence of masses that we read, we can infer later on the exact amino acid sequence from the MS2 data. So this is a very interesting process that has been only recently described by Vicky Wasocki and Bela Pais in two manuscripts. And it is a complicated chemical process where a mobile proton will attack one of these bounds and then cause a break between these two peptide bonds. How this exactly works, it's a semi-stochastic process, so it will not always break in the same way. So for one peptide, it can generate a break over here. The other peptide, it can generate a break at another position. So it depends a little bit. There is some stochasticity involved. Stochasticity, I mean that there is an act of chance involved with where it exactly would break. But since we have many copy numbers of that single selected peptide, we hope that we will for sure break all the possible peptide bonds and have a good measurement at of those masses. Okay, if you want more, read more about this, you can refer to these two manuscripts, or you can just believe me that at the end of the day, if the ion statistics work out well, and we have a little bit luck, that all the possible peptide bonds have been broken a few times so that we can see a good signal in our mass spectrometer. Okay. So how can we now tie this all together? So that's why I come up with an analogy of the marathon. So this is the marathon in London. And you can see there's a lot of people attending every year, and you are a photographer. And the organization asks you, well, I need to have a snapshot of every participant to the London Marathon. So what you could do is you could position yourself at the beginning here, at the start, sorry, and you could try to take a photo of every person that is participating. That would require you quite some time because it's too crowded. But what you could do instead is be smart and position you with your instrument. Finish, because a typical marathon takes you a little bit more than 2 hours to run if you are very fit, and between 2 and 4 hours, let's say. So what you could do is, well, what it will do is it will separate the participants according to their fitness level. In a similar fashion as a mass spectrometer would separate molecular ions based on their mass and charge. So this gives you some opportunity and some time to try to catalog all the participants of the marathon. So how would you do that? Because at the finish, you see there are 3 exit rows. So that's interesting. So what you could do is you take an overall snapshot of this exit row, right? And that would be similar as your MS1 scan. You measure, you look at all the ions that come from the column at a certain time point, right? And you look at the picture because you're very fast, well-experienced photographer. You look at the picture and you figure out which persons you haven't have taken the snapshot. And then you will select the 5 persons which you haven't took a snapshot yet, and you will quickly zoom into those guys and you will take a snapshot of the persons. And you just repeat this process over and over again because you don't want to miss any of the runners that are participating that day. Well, some things can go wrong, obviously. You can well, somebody maybe is very tired and will wander around here in this exit row for a long time, and you will take his, you will see him several times appearing in the overview snapshot in the MS1, and you would perhaps forget that you already taken a zoom in snapshot of this person. So you might decide like, okay, let's take the close-up again. And that close-up, you recognize the analogy perhaps, that is the MS2 scan, yes, that we're taking. So that peptide has been taken a close-up from twice or even 3 times, depending how long he wanders around at the exit. Another situation can happen. A person can keep on running until he is completely out of sight. So while you were taking this close-up, that guy was has passed the exit, and you were never capable of finding him on your overview photograph, and never were capable of selecting him. Or maybe you have seen him, but when the time is there to take the close-up, he may be already run further. So these things can happen also in a mass spectrometer. It's not necessarily so that if a peptide is present, that you also select the peptide for fragmentation. Or if you select the peptide for fragmentation, it could be already gone, resulting into a bad fragmentation spectrum. So keep on in the memory that this is like an ongoing process, lasting for 2 hours, taking photographs. That takes only a few seconds. So that a difference in timing is also the case in LCMS, liquid chromatography, and mass spectrometry. So a mass spectrometer can operate way much faster than the chromatogram. That will take off making a good chromatogram, that will take you 2 hours or sometimes 90 minutes. And the mass spectrometer, that can take these close-ups 10 for every second or even more nowadays. So they can go up to 20, 30, 40 close-ups in a second. This is a slide I got from Lennart Martens from Belgium as well. And this gives you a more serious idea about how the overall shotgun proteomics workflow works. And what we have is we have some cells here that we want to extract the proteins from. So we extract proteins from cells. This is actually most of the work. And then we have a complex protein mixture. So we don't want to analyze proteins. We have been talking about that. We want to analyze proteins by means of their peptides, because peptides are much smaller, much easier to make them linear. They have a way much better behavior on the column and in the mass spectrometer, and it is more homogeneous population. Although there are fractional differences, it is pretty well behaving, opposed to proteins that can come in all sizes, all forms, and it's way more complicated to do proteomics at the level of proteins, although some groups are investigating this as well. But what we do is then we cut that protein with our molecular scissors, we call them enzymes or proteases, to an extremely complicated peptide mixture. So we go from complex protein mixture to an extremely complex peptide mixture. And well, we lost all the connections between the peptides and the proteins. And that is a bad thing, but later on we will try to fix that using bioinformatics. But to make the mass spectrometer capable of analyzing this complex peptide mixture, we need to do separation. And we separate our peptide mixture in less complicated peptides. And then we go to the mass spectrometer analysis. And we take an MS1 snapshot of our data, and we see several peaks appearing, and we let our instrument decide themselves which peaks are worthwhile to further investigate. And typically, the mass spectrometer, they want to have a lot of copy numbers, so they want to have good ion statistics to make such a fragmentation spectrum. Then therefore, it selects usually the most abundant peaks in the spectrum. So here, the mass spectrometer selects 3 peaks, and it will isolate these masses and accumulate them until we have sufficient ions, and then fragment these peptides, and hopefully we will receive a nice peptide ladder. So it means a series of ions, of fragment ions, from which we can infer the amino acid sequence. This process, is called data-dependent acquisition. So we let the mass spectrometer decide which peaks are worthwhile to further investigate. And like I said before, it is a semi-stochastic process. So if I would run exactly the same sample in another run, I could discover some additional proteins and peptides. And perhaps I also could miss out on some proteins and peptides. So there is a stochastic component associated to this type of analysis. Nowadays, a lot of people do not want that because you cannot control what you measure. And that's why there is a new type of analysis, and that's called data-independent acquisition. And that's where we try to measure everything. But the disadvantage here is that we cannot select peptides according to their mass very accurately anymore, but we will select mass regions. But as a consequence of that is that we have some overlapping peptides in a mass region, and we are confronted with a complicated problem. But again, there are bioinformatics solutions to solve that. This lecture, I will just restrict myself to these data-dependent acquisition. So at the end of the day, we have a complicated data set. For example, we were part of a large FP7 project where we collected 131 urinary samples and another 137 urinary samples that we had to measure. This constituted to a total volume of more than 130 gigabytes of data. In total, 600,000 MS1 scans and more than 6 million MS2 scans. So who is going to analyze this data? Well, if you want to do this with humans, impossible. So we need computers to figure out which proteins were present in the sample. The data is also very diverse. So these are all different types of views on a typical LC-MS experiment. And I will just quickly go over those just to name drop. So what you see here at the top, it is not a mass spectrum. No, this is a chromatogram. So this is here a chromatogram. Typically, you can what you see here, these numbers in front. That's time. So this is a chromatogram of a run that lasts for 1 hour approximately. And you see here there is some blank, so no signal. That is what we call the dead time of the column. So it takes a while before peptides start to elute from the column. Very typical for a chromatogram, and then suddenly you see peptide peaks starting to elute from the column. We don't know yet which mass is underneath this chromatographic peak. There could be more even under this peak. We don't know that. That we need to further investigate. But this is how a chromatogram would typically look like. And you can see also very typical, like a heavy bulb of stuff that dilutes at the end of the experiment. That's when we are like rinsing our column with very oily liquid phase, so that we are cleaning and our column from the peptide content. What other spectrum do we have here? Here is a zoom-in scan of a precursor. So this is a precursor scan, an MS1 scan, and we look at an isotope distribution in this graph. So we see here a molecular ion eluting from the column, and on the x-axis we have the master charge, and on the y-axis we have our abundance measure normalized to 100%. We can infer useful information from such an isotope distribution. We can, for example, learn whether a peptide contains sulfur, yes or no, or whether a peptide is really from amino acid. Peptide is composed out of amino acids, but other compounds can also display an isotope distribution, and based on the shape, we could perhaps decide that this is not a peptide, but a contaminant, an other organic form which we should not worry too much about. And you can, from this isotope distribution, you can also infer charge state of the molecule, and when you have the charge state, you can pinpoint exactly the mass value. How you do this, I will not discuss in this lecture. Here you see a typical fragmentation spectrum. So you see a lot of peaks here. These peaks are coming from the same selected peptide, but we have several copy numbers of that peptide, which were broken apart, and you can see all these ions, the fragment ions, they have different masses, and based on these different masses, we can now try to figure out which amino acid sequence we were looking at. And then, not very important or relevant in this lecture, but this is a different type of scan available as well, or as part of a spectrum is a better way of saying, is available in the data. And this is the reporter ion region. When you want to do quantitative proteomics, there is something called TMT or iTrack. You can label the samples with isobaric tags and then do a multiplexing where you combine several of the samples in one pool and then analyze the pool. And in the mass spectrometer, you will demultiplex that information again. And when you demultiplex that information, you will get a reporter ion region that tells you what is the proportion of that peptide in that sample. And you can see, for example, in sample 129, which is labeled by the label 129, that that peptide is way more abundant in that sample than it is in the other ones. So different types of data, complicated structure. How are we going to analyze this? And well, we're going to use this using computers. So shotgun proteomics, also known as gel-free proteomics or polymer proteomics or peptide-centric proteomics. There are two big steps. So we have a disassembly step where we destruct the proteins into peptides into fragment ions. And then we have, that's what we call the disassembly step. And that's how we generate the data. We do that in the lab. And then we have a reassembly step. And that we do on the computer. We will try to figure out, looking at these masses of the fragment ions, which peptide sequence we are looking at. From that, we will infer which peptides we're having in our sample. And from the peptides, we go back to the proteins. So there's a disassembly in the lab, reassembly on the computer. So generating the data, interpreting the data, and I see actually a nice analogy with next generation sequencing. We also do that, so we break the DNA or RNA sequence completely apart into our lab and next-gen sequencer, and then afterwards we will try to reassemble the genetic information using bioinformatics. One important term that I want to still mention on this slide is the term peptide spectrum match, or abbreviated PSM. So when we attach a certain amino acid sequence to a tandem mass spectrum, we call this a peptide spectrum match. That's something that I want you to recall for later. So let's have a closer look at all these disruption steps, and I have some limited time today, so probably I will stop this discussion after the disruption steps and continue my talk in the third session. So what are the disruption steps in proteomics, and why is it important that I mention this here? Well, first disruption step is if we want to digest protein, which is a three-dimensional nanomachine, very compacted globular molecule. Well, what we need to do is to unfold that molecule. And we have technologies to unfold that. We can, for example, use SDS and heat up the protein like the same thing you would do if you cook an egg. So if you cook an egg, it will start to denaturate. That's what you want to have. You want to denaturate a protein. But there are some connections in that protein that are very firm and strong. And that's what we call disulfide bridges. So these disulfide bridges, we need to oxidize those disulfide bridges. We need to break them. But hey, it's nature. So if you break them and you leave them back in solution unchanged, they will form the disulfide bridges by themselves. It's self-assembling. Perhaps in the wrong way, but again, you're there with these firm bonds. So we don't want that. So once we have oxidized these disulfide bridges, we need to modify the sulfurs to prevent that they can be formed back these strong bridges. And we do that by a modification called carbamidomethylation. And it is important to realize if you modify your protein a little bit with a chemical compound, you will change the mass. So that's why I'm telling you this. So every step that is undertaken in while processing the proteins that could alter the mass, you should be, as a bioinformatician, be aware of, because you will need to adjust your search engines and your algorithms so that it can accommodate the change of mass. That is very important. That's why I'm telling you these description steps. And although it's chemical, it's important to realize for a bioinformatician. And well, good. Once we have denatured, linearized our molecule again, it's open for digestion. So we will digest the protein by proteases or enzymes, and we cut that protein into peptides. Typically, for proteomics, most of the time we use trypsin, and that is very convenient. Trypsin gives us a basic amino acids to the N-terminus and the C-terminus of the peptide. But we will come back later to that. And then, obviously, when we have the peptides and we separate the peptides and have them in the mass spectrometer, as a final step, we will blow them to smithereens just to investigate their fragment masses. And we do that in this talk by collision-induced dissociation and combined with the data-dependent acquisition. Okay, so I think before we go any further into the nice details of how we now could develop a bioinformatics algorithm to analyze this data, we will stop this session because I will definitely need 1 more hour to explain about bioinformatics here. Hello, welcome in this third session on a general bottom-up proteomics workflow. In the previous sessions, we talked a little bit about the context, and in this third session, I want to go into more detail on the bioinformatics. Remember that in the lab, when we are trying to identify proteins, these proteins undergo a disassembly process. So we have a mixture of proteins. These proteins will be denatured, so they will be linearized. We will cut these proteins in peptides, smaller pieces of amino acids, that is to say, so that they behave more stable and are more homogenic mixture. And then we will further analyze these peptides using liquid chromatography to reduce the complexity of the sample and mass spectrometry. And in this mass spectrometry, we collect MS1 data. That means we catalog all the peptides that elute from a column at a certain time, and we record many information of these peptides. And then in the MS2 phase, we will select one of these peptides and we will further break them down into a tandem mass spectrum. And in a typical proteomics experiment, we have thousands of these tandem mass spectra that needs interpretation. And we will use that, a computer for that. And that's why we need to talk about bioinformatics in this session. So how will the bioinformatics algorithm look like? So what we will do is we completely mimic the procedure that we do in the lab. We do that also on the computer. And first thing what we know about our sample is which organism it is. So what we will do is we will take a FASTA database that contains all the known protein sequences of that organism. Typically, we will limit ourselves to canonical protein sequences. That means that these are the protein sequences that are most commonly agreed upon by scientists. Special isoforms or mutations that a particular individual has, these sequences are not included in such a FASTA database. And therefore, our bioinformatics algorithm is blind for these findings. So good, once we have our organism FASTA database, then we will cut these proteins by trypsin, and such that we have a peptide sequence database. And then we will filter down these peptide sequences so that we have a list of candidates. And these candidates will then be fragmented also on the computer.
so that we have virtual fragmentation spectra, and we will compare these virtual fragmentation spectra with our actual tandem mass spectrum, and we will compute a goodness of fit score. And if this goodness of fit score is above a certain threshold, we say that seems a pretty good peptide to spectrum match, and we call that tandem mass spectrum to be associated with a certain peptide sequence. So don't worry if you don't understand all these steps I just mentioned. I will go over them individually in the next slides. So first of all, we need to do proteolysis or cut our protein with proteases. And I told you in the last session, the most important protease in shotgun proteomics or bottom-up proteomics is trypsin. And trypsin allows us to cut on arginine or lysine, which is the amino one-letter code R or K. And if we do that, we ensure that there is a basic amino acid at the C-terminus of the protein. And that is pretty interesting because that basic term will most likely attract a proton during the ionization, making us to have a charged C term. And we also already have a basic amino term, N-terminus. So that one will also most likely attract an additional charge. So we have doubly charged molecules most of the time when we do electrospray ionization in combination with trypsin digestion. And that's a good thing, as we will see later on when I'll talk about fragmentation spectra. So what trypsin does in real life, typically we put trypsin to our protein sample and let it rest overnight at 37 degrees Celsius and let it do its job. Trypsin is not perfect and will once in a while miss cleavage, and that happens. So we need to accommodate that behavior also in the computer. But what we will do on the computer first is simply cut the proteins into peptides by cutting on the arginine and lysine, except when there is a proline at the C-terminus site. Then this trypsin will not cut. And we have like a regular expression for displaying the behavior of our protease, and in this case, we use trypsin. So skip this slide. Good. So what can happen in real life is that our peptide or protein has been processed already in our body, or maybe has degraded a little bit after we were extracting the protein. So it's possible that we have semi-tryptic fragments. It's also possible that our trypsin will miss a cleavage, and then we have still an intact arginine or lysine here in our peptides sitting, and we can accommodate for that. So typically, we need to specify in the search whether we want to also consider miscleavages. That's easy. The other ones, like semi-tryptic peptides, that is more complicated. That is a field of interest of the science called Proteomics that will look into which proteases are active in a certain biological situation and what can be learned from that. But we will not discuss this in this session. Good. So once we have our database now with peptides, which potentially could have missed cleavages, we need to apply a peptide mass filter. And that's exactly what happens also in the mass spectrometry. Recall that I mentioned that this MS1 layer catalogs all the peptides that are coming from the column at a certain retention time. And the mass spectrometer will select only a single peptide for further inspection during tandem mass spectrometry or during the MS2 step. We can mimic that on the computer as well, because we know the mass of the peptide that is being fragmented. So it is of no use to us to consider all the peptides in our peptide database as a candidate for that spectrum. No, we can limit our search, and that's interesting. So we could, given the error tolerance of our mass spectrometer, we can ask the computer to give us all the peptides back of that parent peptide, the parent mass of the peptide, plus minus this error tolerance, and then we will have a limited list of candidate peptides that we need to consider or observe the tandem MS spectrum. Now, I stressed in previous session that all possible biological modifications that can occur in biology or chemical modifications that could occur during processing of the sample, we need to be aware of that. Because if there is a modification, that modification can alter the mass. And if it alters the mass, then we are clearly focusing here on a different region in our peptide database. So we need to take every possible modification into consideration. For example, if we know that we have reduced our cysteines with carbamidemethylation, this will add approximately 57 Dalton to the peptide mass. So if we believe that a certain spectrum could be the result from a peptide that contains a cysteine, then we first need to subtract 57 Dalton from that mass and then filter our peptide database to see if there are peptides that contain cysteine in that region and consider those peptides as well. So you see, we double here the search space of we need to search our peptide database twice, once for the peptides that do not contain cysteines and once for the peptides that do contain cysteines, because we do a chemical modification of cysteine in the lab. But that's still easy. Imagine that we have unknown biological modifications like phosphorylation. So a peptide might be phosphorylated or not. We call those modifications variable modifications, because we need to be open for both types, modified or not modified. And potentially, there can be more modifications. For example, like acetylation of lysines or N-termini, or oxidation of the methionines, and they can happen or not happen. And to consider all possible combinations, exploding our search space. So that's why open modification search algorithms are being developed as we speak, and it's still a topic for research. Good. So once we have now selected our peptides, or candidate peptides that we need to map on our spectrum, that's a little bit tricky because peptide is just a concatenation of letters, amino acid letters. So it lives in the text world. And our tandem MS spectrum is just a bunch of peaks. It's a spectrum. It lives in the spectrum world. So how will we now try to figure out whether a piece of text will match spectrum? And this is the here of what we will do is we will create a virtual spectrum based on the text, the peptide sequence. How we will do that when we do CID fragmentation. We know very well how this works, and CID fragmentation will further disassembly a peptide sequence, but this time in the gas phase, and it will break the peptide bonds. So it will break peptide bonds, and doing so, it will form B ions and Y ions. So if we break a peptide, so this is here a peptide composing out of 4 amino acids. If we break this one at this position, we will create B1 fragment ion and a Y3 fragment ion. Why would we call it B1? Well, we break something in two. There is a part that is related to the N-terminus of the peptide, and there is a part that relates to the C-terminus of the peptide. So Y ions are related to the C term, B ions are related to the N term, and the numbers, they express the position of the amino acid in the peptide, going from left to right if we talk about B ions, going from right to left if we talk about Y ions. Here I need to talk again about the benefit of using trypsin, because we have here now arginine or a lysine sitting on this C-terminal side. Most likely the C-terminus will get a charge, and or N-terminus will get a charge. So if we break something in two, right, we will most likely see the B ion appearing and the Y ion appearing. And that's a good thing. If this guy was only carrying one charge, then it would be based on coincidence if we would see the N-termini or the Y or the C-termini. So then we would only see one of the fragment ions. But when there are multiple charges present, it can also be triply charged, obviously, then we will see, in most cases, the N-termini and the C-termini. And that's good, because what we are playing here is the population game. We don't fragment one of these guys. No, we will have like thousands of these peptides that are being fragmented. And yes, there is a propensity for a proton to attack a bind and will fragment at a certain position, but it's also a semi-stochastic process behind this. So if we are being to fragment this peptide 1,000 times, then a few times this peptide bound will be broken. Another time this peptide will be broken. This peptide bound will be broken, and yet another event this peptide bound will be broken. So hopefully we will see a nice sequence of masses from which we can derive then the amino acids. So this is how now we will generate, for every candidate peptide, we will now generate a virtual spectrum by enlisting all the B ions and all the Y ions, taking into consideration possible charge states. Typically, we will not care too much about intensities in the classical database searches. Nowadays, new AI tools are available that are able to accurately predict the fragment intensities of these peptides. But in the past, using the simple classical search engines like Mascot and Sequest, we typically considered an intensity of 1, or just considered that the trypsin-based Y ions are just a bit larger than the double as large as the B ions. And obviously, we don't know which parts will be charged. So when we have one or doubly charged peptides, we will usually compute 1+ fragment ions. From the moment that we have triply charged peptides, we will also start to predict doubly charged fragment ions. So increasing the number of B and Y ions, and also increasing the possibility of false chance findings. Good. So this is a little bit complicated slide, but it tells us just how we go from peptide sequence to virtual fragmentation spectrum. And this is, for example, an online tool in which you insert a certain peptide sequence and what you will get here. So I asked this tool to fragment this peptide based on for singly charged fragment ions, and we have here just a list of B and Y ions of masses. And what we need to do now is take these masses and see if we find corresponding peaks in a certain spectrum. And we can do that, for example. So we will, in this spectrum, we will see a Y6, Y7, and a Y8 ion corresponding to this peptide. We see some B ions, And for that reason, we can conclude that the spectrum that we're looking at indeed is generated by fragmenting this peptide sequence. So it's matching the masses of between an observed spectrum and a theoretical spectrum and computing a goodness of fit score that will lead to a positive identification of a fragment spectrum. I will have time for one more example. For example, here is another peptide sequence that for which I generated the P and Y ions. And indeed, I could nicely annotate some ions. And you can already see that chemistry is at work here. And we have some neutral loss of water from a peptide generated an additional peak that I cannot just explain by only looking at these B and Y ions. So you understand that some ambiguity and some to fully interpret a mass spectrum. So I like this example a lot, but like I said, there is some ambiguity because another explanation for this fragment spectrum could be this sequence. So if I would generate B and Y ions for this sequence, then I also can see some B ions and Y ions and conclude that this spectrum is not from the first peptide, but from the second peptide. So you see this mass matching when you want to do a peptide to spectrum match. It's a good idea, it's the best that we have, but there is some ambiguity present. And luckily today we have peak intensities, fragment peak intensities that come to help to reduce this ambiguity. All right, some 2 more minutes left before I will conclude this session. The two archetypes of search engines that are very popular in the past are Sequest. I will not go into detail, and Mascot. So these were mainly used in the lab. Sequest is still the core engine for the proteome discovery workflow, and Mascot still is in business. So I assume that still labs are relying on Mascot searches for their spectral identification. This is an old reference, 2010, but back then there was a long list of search engines. I can only say that the list has become longer now, especially with these new AI tools. So there are a lot of search engines that you can try to do peptide to spectrum matching. The bad thing is they don't agree all. So if you use multiple search engines, you will find that they will complement each other in their findings. So I want to conclude this third session here. And next session, I would like to talk a little bit about how we build confidence now in this finding, because we have now a goodness of fit score. But what does this score mean? When can we be confident about that score? And that will be a topic for the next session.
Okay, welcome back in this fourth session on search engines for mass spectrometry-based proteomics. In the last sessions, we have seen the context of proteomics. We have seen the technology that is used to analyze proteins using liquid chromatography and mass spectrometry. It ends up with a huge pile of data, and we have seen some bioinformatic solutions to analyze and blow through this data. As a conclusion of last session, we have a list of various search algorithms. There are many, but the basic principles, they hold and were explained in the previous session. The question now is, how will we homogenize results across all these different search engines? Because they all provide score, but we don't know if a score is a significant bona fide peptide or maybe just a chance finding. So how do we control our false positives with this technology? And I will spend this session to discuss a strategy that is omnipresently used in this type of search engines. And it's about peptide confidence. So to reiterate, we have a peptide in our sample. We do tandem MS on this peptide, and we've got amino acid ladder or fragmentation pattern we see, because we have not only one such peptide, we have several copy numbers of this peptide, and the propensity to break is well, there is a stochastic process. So once in a while, this peptide will break in two parts at different cuts, and as such, we will see a peptide ladder appear, and we call that B ion series and a Y ion series. So we use now, we mimic that entire process that we've done in the lab. We do that on the computer. So we have a protein sequence database, which we turned into a peptide database, and then we compare the acquired spectrum with a theoretical spectrum. And when our goodness of fit measure that we use gives a high score, then we say this is a confident finding. So as a result, we have often more than one candidate peptide that we want to verify on our required spectrum. So as a result, we have a list of peptides with scores. And we can rank these peptides according to the scores. But what should we now trust? Maybe this 3.3 is indeed a bona fide finding, but it might be possible that there are two peptides in this acquired spectrum, and that this second peptide should also be considered. Or maybe the score of 3.3, this is indeed the highest ranking peptide, but maybe 3.3 is not good enough as an explanation for this spectrum. So how could we now actually distinguish a true from a false identification? And this method is almost always used in database search engines, and it's called a target-decoy approach. So what we will try to do is to get an idea about the null hypothesis. How would a spectrum match on a random candidate peptide? And if we do that many, many times, so we know that this is for sure a fake random peptide, then we've got an idea about this score is distributed. But it's a complicated procedure, and it comes with a lot of problems. So how random should such a peptide become? And to remediate that, the proteomics community invented this target decoy approach. And the idea is, well, we have our target database. This is the protein. This is the peptide database coming from our protein FASTA file that we have downloaded. But we will also generate a decoy database, a database with fake sequences. So how do we generate these fake sequences? Well, we will reverse our original protein database. So we swap the N terminal and the C terminal, and then we will do a similar digest as we did on our target database. As such, we have a decoy database with nonsense sequences. And we concatenate these two databases and we treat them as one big database. So now we will have in our experiment, we have N spectra that we have acquired from our sample, and we will analyze each spectrum using this database searching. And a difference here is we will only consider for now the first ranked peptide. So we will only we will only annotate or allocate a sequence to a spectrum for the highest ranked peptide candidate. So that is the procedure. So we don't consider any more rank 2, rank 3, or rank 4 peptides at this moment. So what happens is A sequence can now come from the target database or from the decoy database. And possibly we will assign a sequence. We will record where it came from. So for example, my first spectrum, we could annotate a sequence to that and with a score of 4.5. And that sequence is coming from our target database. And our second spectrum, we also did a search on this concatenated target decoy database, and we would assign again a sequence with a score of 3.9 that also came from the target database. And so we will we have ranked our list, and we can scroll down this score. And at some point, when the score is becoming lower and lower, so it means that we're getting less and less goodness of fit between the acquired spectrum and the theoretical spectrum. So at some point, we see that decoys sequences creep into this list. And that is bad news, because the decoys, well, we know that these are false sequences. They do not exist. So if we have 1% of decoys, so how far do we drop in this score? Well, we lower this red line here, so we will apply a cutoff on the score at that position, where we have 1% decoys, up to the moment that we have 1% decoys in our list. These decoys are nonsense sequences, so they are for sure false positives. So by this procedure, then we can then argue that we allow 1% of false positive findings. So this is how the reversed target decoy approach works. It is a very simple and effective method, and it allows us to estimate the error rates for an entire data set as a function of this filtering threshold. Disadvantage is we don't gain a confidence level for individual PSMs, but that's okay. The good thing is of this target decoy approach is that we can compare across different labs, across different search engines, using the same reversed target decoy approach. What we always can do is also use additional information to gain confidence about our findings. And there is what happened here? Okay. There is a lot of additional information that we can add in, but I will not discuss this in this session. Just to mention a few, we could, for example, look at the mass accuracy, or we could look at how many miscleavages we observe. So peptides that have a low confidence with a lot of miscleavages, that might be a sign that this is a false finding. Or, for example, if we discover a peptide and we could compute for that peptide the isoelectric point, so that is a physical chemical characteristic of a peptide, and it is related to the hydrophobicity of that peptide. So if we then could relate that to the retention time, we could try to understand whether this finding of this peptide would indeed elute at a certain time from the column. So there are a lot of additional filtering steps that could be used to further fine-tune our results. So by this, you already can see that it's a very lossy technique, and that's why we also sometimes call this shotgun proteomics, because we want to identify proteins, and we'll try to measure as many of peptides related to that protein in the hope that at least a few of these peptides of that protein are identifiable by mass spectrometry. But in the meantime, we will not, we will throw away a lot of information and filter out a lot of data. That's why we need better algorithms to search the data sets. Another interesting part of additional information is, for example, if we will receive a sequence from our database search, what we can do is compute mass value of that peptide and compare it with the actual observed mass value. And if we would do that for all our peptide to spectrum matches, then we will receive a plot that looks more or less similar as the plot here on this slide. So what we will see, what you can see from this slide, is that in our search, we allow an error tolerance of 10 ppm. On our mass spectrometer, 10 ppm, that's a lot of error tolerance. And you can see that the sequences that are falling at the neighborhood of the -10 or +10 parts per million ppm, they all have low scores. But if we go more to the 0 ppm, we see a spike up of scores. So that means that this spike up means that these peptide spectrum matches receive higher scores and therefore are more confident. And they are closer to the actual mass of the observed peptide. What we can see, in fact, in this plot is even we can see that there is a miscalibration. Ideally, this spike of identifications should be centered around the 0 line here, but that's not the case, and therefore we can immediately see that the instrument is probably a little bit ill-calibrated and requires a new calibration to put this spike up cloud back into the middle, centered around the 0 value. Okay, so what is then a typical result of such a database search? Well, since we can allow more peptide to spectrum matches than only the first ranked, it's possible that the number of identifications outrank the number of spectra that we have. Sometimes we also need to identify a spectrum multiple times because we don't know exactly the charge state of the spectrum. So it could be a spectrum related to a peptide of mass 1000, or maybe because I don't know the charge mass around 500, and I need to search multiple times. And it's possible that each search provides me a successful identification. So it's well possible that we have a long list of peptide identifications. And what I haven't been talking about so far, and I could spend an entire session on that discussion, is, well, we're not really interested in finding peptides in our proteomics approach. We are interested in finding the proteins, the gene products. So we do protein identification based on peptide homology. And how this works is presented on the following slide here. What we see is we need to infer which proteins we now have actually observed. And what can happen is if we digest protein, it results into a number of peptides, but some peptides can be shared across multiple proteins. So how will we do the inference of the proteins based on the peptides? And there are several strategies that you could use. You could come up with a minimum set. So, for example, a minimum set of proteins that explains the observed peptides. You could also come up with the maximum set and say that, oh, every protein that could be explained by this set of peptides should be in my list of discovered proteins. So there are multiple approaches. Typically, it boils down to solving some type of customer satisfaction problem, a complicated problem to balance out to figure out what is the list of most probable proteins that could give rise to this selection of peptides. So very interesting procedure possible in protein inference, but you can attend an entire course on this subject alone. I want to conclude this session again with a few slides. This one is an interesting slide I got from Lennart Martens from Ghent. What should we learn if we do database searching? Well, we try to automate a lot of our work and try to interpret our data in an automated way. This workflow makes a little bit abstraction of the spectra, so we don't look at the spectra anymore. We look at the identifications and the proteins found. So generally, when you use automated workflows, the idea is don't simply trust computers. Automatic software here is this is a picture from Japanese scouts during an international jamboree in the Netherlands. It says possibly, I don't know. I don't think we have these mountains in the Netherlands. So it is complicated work. So what you can see, the search engines generally they work well, but we match peaks. And here what you see here is an isotope pattern of a fragment ion, and here the second peak is annotated as a Y4 ion. So you really need to figure out and look at the annotated spectrum to assess the quality of your data. But obviously, it is almost impossible to do this, given that you generate gigabytes of data in a single experiment. So still, if you want to gain real confidence about a peptide spectrum match, best is to look at the spectrum. So the take-home message here is, well, proteomics and mass spectrometry, that is not plug-and-play technology. It requires quite some investment to understand the technology and be capable of interpreting all these results. So this concludes this fourth session on how we get confidence about our findings. Take home is, well, don't trust the results blindly, always further investigate. Especially when you think something weird is going on or an off conclusion is made, then you definitely have to go back to the data and look at the annotations. This is what I've explained now is the methods from like used a decade ago. Now with the rise of AI technologies, there are, especially with the rise of generative AI, there is now a new generation of algorithms that that make use of better models that predict the theoretical spectra. And because they now not only predict the mass values of these theoretical spectra, but also can predict intensity values very accurately, we can use that information to come up with new goodness of fit statistics and have a better, more stringent way of deciding whether something is a peptide or a correct peptide sequence or false peptide sequence. So again, with this additional information, we now have intensity of the fragment ions that that come to work and will tremendously help us to set up more stringent searches. If we have a stringent search, we can also open up our search space and we can start to ask ourselves the question, do we still have to work with these canonical sequences? Maybe because if there is biological variation, it's possible that there is a mutation in a certain protein, and we can now, with this stringent algorithm, we can open up our search space and we can start to find out what the mutation or which or whether there are mutated peptides present in a certain protein. And such mutations, again, are obviously predictive, or could become predictive to identify the human from who we derived the sample. So in the past, we used canonical sequences, and we were not able to, or bioinformatics methods were not able to identify these subtle peptide mutations in an untargeted way. Now we arrive at the technology where we are capable of figuring out that information, bringing up an entire series of questions about ethics and proteomics.